Hello, everyone, and welcome to today's live broadcast, TCR sequencing and BCR sequencing solutions, applications to clinical research projects. Today's webinar will be presented to you by Dr. Brian O'Connor, Associate Professor and Scientific Director of Genomics at National Jewish Health. I'm Christy Jewell of Labroots, and I'll be moderating today's event. Today's educational web seminar is brought to you by Labroots and sponsored by Thermo Fisher Scientific. For more information on our sponsor, please click on the Thermo Fisher logo on the left side of your screen. Before we begin, I would like to encourage you to get the most out of today's webinar by submitting as many questions as you want at any time you want during the presentation. Simply type them into the Ask a Question box on the left and click Send. We will answer as many of your questions as we have time for at the end of the presentation. If you have any trouble seeing or hearing the presentation, click on the Support tab found at the top right of your presentation window, or use that Ask a Question box on the left. Today's presentation is educational and thus offers continuing education credits. Following this presentation, those credits can be obtained by clicking on the Continuing Education Credits tab at the top right of your presentation window and follow the process to obtain your credits. I would now like to introduce today's speaker, Dr. Brian O'Connor. Dr. O'Connor is an Associate Professor and Scientific Director of Genomics at National Jewish Health in the Center for Genes, Environment, and Health. His work focuses on understanding how epigenetic mechanisms regulate the decision processes governing immune cell activity in the context of disease. For a complete biography on our speaker, please visit the Biography tab at the top of your screen. Welcome, Dr. O'Connor. You may now begin your presentation.
Okay, thank you for that introduction. My name is Brian O'Connor. I am an associate professor at National Jewish Health, and I've been the scientific director of our genomics facility for a couple of years now. Our facility has a lot of experience working with Thermo Fisher and their ion AmpliSeq products. And today what I'm going to be talking about is their TCR-seq and BCR-seq solutions, specifically in the context of how you can use these tools for clinical research projects. So National Jewish Health, we have a lot of experience looking at TCR. Many of you in the audience may know of Pippa Merrick and her husband, John Kapler. They've been looking at T cells and specifically T cell receptors for quite a long time. And using next-gen sequencing approaches, Pippa's been looking at TCR usage in T cell populations using next-gen sequencing. For example, before 2013, she was using the 454 platform. Around 2013, working with Thermo Fisher, we came up with a homebrew system to look at TCR sequencing using the ion approach. And in 2019, Thermo launched their kit-based solutions for TCR and BCR sequencing. We became a preferred academic partner of Thermo Fisher for TCR and BCR sequencing. So we have a long history of using ion AmpliSeq and ion approaches for different questions at National Jewish. So for today's talk, what I'm going to be giving you is a brief overview of the TCR sequencing and BCR sequencing solutions that Thermo Fisher provides. And then I want to give you a brief overview of how you can apply these tools to specific kinds of questions in inflammatory diseases for clinical research. So I'm going to give you two different brief overviews of case studies, one dealing with a disease called sarcoidosis and another one called cystic fibrosis. And of course, today, given everything that's happening in the world, it would be remiss if we didn't briefly discuss COVID-2 applications for TCR and BCR sequencing as well. So talk briefly about that. There's obvious examples of how to use TCR sequencing and BCR sequencing in immunobiology for things like animal models. And we'll use a future webinar to discuss those applications. So a brief overview of TCR and BCR sequencing and why you might do it. So these are some slides that Thermo Fisher was good enough to provide me with. So when thinking about the adaptive immune system, there's really two major cell types, T cells and B cells. And one of the things that makes T cells and B cells special is this idea of combinatorial diversity. So you have V D, and J genes that recombine at a DNA level. These recombination events lead to unique combinations that, when translated into proteins, become the B cell receptor and the T cell receptor that are expressed on the cell surface of B cells and T cells. The BCR and TCR enable B cells and T cells to see antigen, to recognize pathogens. And when B cells and T cells recognize these pathogens, they undergo rapid proliferation in a process called clonal expansion. This allows the immune system to deal with an invading pathogen or deal with some antigens. In this process, that unique combination of VDJ gets amplified within the cellular population. So in one way, in thinking about this, the clonal expansion process identifies an antigen response, and that antigen response is due to that unique gene sequence. So if you had a way to see that unique gene sequence, to read it and to track it, it would enable you to track the immune response and to identify an ongoing immune response. So in terms of clinical research, when thinking about acute responses or chronic diseases, understanding when an immune response is occurring and what kind of an immune response is occurring becomes a really powerful tool. So sequencing the unique combination of VDJ becomes a really great way to track an immune response. Historically, there's been lots of different methods to look at TCRs and BCRs within T cells and B cells. So people have used things like 5' race or multiplex PCR. These methods have advantages and disadvantages. In the context of 5' race, it's a great technique, but it's not very scalable. You can't really apply it to lots of different samples, or at least it's very difficult to do that. For multiplex PCR, you can look at lots of different variable regions at the same time, but because of the differential combinations of lots of different primer pairs, you start to get into some issues with loss of certain transcripts. And so they have their advantages and disadvantages. Ion AmpliSeq provides a really great solution to some of these advantages and disadvantages. Essentially, this is a combination of PCR and next-gen sequencing. So use PCR to target the region that you're interested in this case, the T cell receptor genes or the B cell receptor genes. Those targets are amplified, and then those amplicons are made into a next-gen sequencing library, and the sequencing platform then reads off those transcripts, amplifying that signal so you can read it pretty easily. In terms of the sequencing platform, Thermo Fisher has a flexible system. They have different kinds of sequencers in terms of their capacity, going from an S5 to an S5 plus to an S5 prime. National Jewish Health, we use an S5 prime all the time. And they also have different size chips where your samples are loaded onto the sequencing platform. And the chips also come in different sizes. So again, you have that flexibility. You can put different kinds of samples on there, different numbers of samples. And really, I think one of the huge advantages of this entire system is not just the combination of the way the assay works, this combination of PCR and NGS, but it's also the analysis software on the back end. It really, at least for us, has become a game changer. So Thermo Fisher's made it very easy for researchers to analyze their own data through this software suite, this suite of tools that is easily accessible in the cloud. So it's called their Ion Reporter software. So they have lots of different tools on there. You can apply it in lots of different contexts, not just immunology, but they have things for exomes and metagenomics. We've used a lot of these tools in our research here at National Jewish. And specifically in the context of TCR and BCR repertoires, a lot of this types of questions or types of analysis that you might want to do, whether it's just looking at the VDJ sequences or trying to track clonal expansions, or in the context of T cells, looking at convergence of different T cell receptors, or in the context of B cells, looking at somatic hypermutation analysis or lineage analysis. All of this is really easily done on the Ion Reporter system. It's very intuitive, very easy to use. I have a PhD in immunology. I do not have formal training in bioinformatics or computational biology. And for me to be able to go on to the system and use it and create graphs that are publication quality or able to stick into a grant, it really has been a game changer for us. This assay can use either DNA or RNA as an input. There's advantage and disadvantage to both. If you are a researcher that has a lot of FFPE type samples where you know the material is going to be fragmented, DNA is probably the way to go for that. And there's both a long read and a short read version of this assay, so the short read version is probably the way to go. On the other hand, if you are dealing with a specialized cell type where you know you're not going to have a lot of cells or the ability to isolate a lot of cells, RNA is going to be the way to go. Again, because of that expression is going to amplify the signal. So there's reasons to use either DNA or RNA, and we can help discuss which one to go for specific projects. And then again, because TCR and BCR sequencing becomes a great tool, a great window into an immune response, when thinking about clinical research, it allows you to ask lots of different questions. So in terms of malignancies or autoimmunity or vaccine immunology, it allows you to think about what's happening within the immune response and to track that response over time. Bulk sequencing, which is what we're talking about here, versus single cell sequencing, answers different kinds of questions. But really, when thinking about clinical research settings where you are going to have to look at lots of different types of samples and different numbers of samples, bulk sequencing becomes a lot easier just in terms of price and the practical ability to apply it to lots of samples. So there really are a lot of different available tools within the AmpliSeq family, and so we at National Jewish have used a lot of these products and have had really good experiences with a lot of them. So I encourage anybody to look into all of the different possibilities that Thermo has provided. So with that, I want to start talking about the different applications or different kinds of questions you can ask with TCR sequencing and BCR sequencing. So I'm going to give you two different case studies that we've been performing here at National Jewish, two different research stories. So this first story is a project that I've been involved with Lisa Meyer, who's an MD at National Jewish Health. She's a specialist in the area of sarcoidosis. And we've been looking at the role of T cells in the context of this disease for a number of years now. So sarcoidosis is an inflammatory chronic disease. It's one of the hallmarks of this disease are these granuloma formations, which are groups of immune cells like macrophages and T cells and B cells that come together and form this inflammatory component. One of the issues with sarcoidosis when thinking about how do we understand it, it's really a disease of unknown etiology, so we don't really understand why this disease starts. Different kinds of infectious agents or organic particles or inorganic material are thought to contribute to inflammatory reactions that become chronic over time. There's a variable disease course, so we know that CD4 positive T cells are involved in this disease because we see them in the inflammatory spaces. But from one example to another, there's really a variable disease course, and so we have a tough time understanding why different inflammatory situations occur. And of course, because there's a variable disease course, there's also variable progression. So in some instances we get remission, in some instances we get progression, and progression leads to this fibrotic type of reaction that occurs within the lungs, leading to worsening lung function. So be really one of the key things that we need to understand is what's happening in the context of this immune response, tracking the immune response and understanding the programming that's occurring. So with Lisa and others at other institutions, we've been tracking T cells in the context of sarcoidosis for a while now, and some of this work was supported by NRL1 from NHLBI. So our main one of the main things that we're trying to understand in this context is when you think about chronic diseases, mature T cells that can exist in your body for a long period of time in the context of these chronic inflammatory conditions are going to experience lots of signals that are derived either from the disease state, from the microenvironment, from genetic programming. And all of this comes together in the context of this huge combinatorial diversity in the context of specific T cell receptors, and again, also in the context of B cells with B cell receptors. And over time, this can lead to changes in the population of T cells or B cells. And so what we'd like to understand is how does that molecular programming that's occurring contribute to the pathogenic activity of the immune response? So in the case of sarcoidosis, we know that Th1 type of activities in CD4 positive T cells contributes to disease. But we'd also like to understand what are the molecular programs that might drive more T regulatory cells in the same component, in the same population of CD4 positive T cells that would be more protective. So if we could understand that decision process and those signals that are involved in driving different types of T cell function, we could then understand better what's happening in this disease. So one of the first things we were able to do was to take CD4 positive T cells derived from peripheral blood cells and perform RNA-seq. So what you're seeing here is unsupervised clustering of differentially expressed genes comparing sarcoidosis samples versus normal healthy samples. So the sarcoidosis samples are denoted in the red bar and the normal samples in the blue bar. So one of the gratifying things that we first saw at this really global level is that indeed the normal samples cluster separately from the sarcoidosis samples at a transcriptional level. When we performed IPA pathway analysis, we were able to identify immune pathways that are specific to sarcoidosis samples versus control samples. And again, what we saw were some of these different T cell functions that were popping out. So Th1 and Th2 activation pathways, or Th2 specific or Th1 specific pathways. And so it was telling us that in the context of sarcoidosis, the CD4 positive T cell activity and function is being differentially regulated. And so next we wanted to ask the question, well, if we think about sarcoidosis and different disease phenotypes, so different types of disease in terms of severity or the phenotype of what's happening in those different types of sarcoidosis, we wanted to understand, could we see RNA signatures that distinguish those different disease phenotypes? So here what you're seeing, again, is unsupervised clustering of differentially expressed genes from RNA-seq of CD4 positive T cells from the peripheral blood. And we do, again, see differential clustering of different phenotypes. So the least severe phenotype, SARC0, is in blue. An more inflammatory type of sarcoidosis, SARC2, is in red. And then SARC3 is a little bit more of a fibrotic type of disease, and that's denoted in green. So each of the different phenotypes are differentially clustering. And the bottom line here is when we looked at the different genes that are hallmarking or denoting the different types of phenotypes, we were starting to see the emergence of specific functions, at least at a transcriptional level. So when thinking globally about sarcoidosis versus wild type, we started to see Th1 versus Th17. When we started to delve deeper into the specific disease phenotypes, we started to see the emergence of functions like Th2 or TFH. And so this led us to really come to an understanding that there are different T cell programs that are being regulated depending on the different disease phenotypes. And so this led us to really think about, okay, what is happening within the immune system? There seems to be differential regulation occurring. And so this really leads into then TCR sequencing. What we were looking for was some biomarker, some handle on trying to understand why you get different disease phenotypes, why disease progresses variably from one instance versus another. And so what we wanted to ask the question was, within the CD4 positive T cell compartment, if we use TCR sequencing, can we identify different types of inflammation that are occurring based on different disease phenotypes. So what I'm going to show you now is TCR sequencing data that we were able to get from the Ion AmpliSeq tool, from the sequencing kit. And this is data that was directly generated from the online tools that Thermo provides. So what you're seeing here is a graph for what is called evenness. So evenness is an idea that if you see no clonal expansion across all of your T cell population, you expect the representation of your TCR genes to be completely even, meaning that there's no bias in the usage of those different genes. So evenness on the top right, you're seeing a scale from 0 to 1, dark blue being the most even, white being the most uneven. So blue basically means no clonal expansion, white means clonal expansion. The way this graph is laid out is along the x-axis. On the top, you see the different V genes, and on the y-axis is the CDR3 length. So each individual dot or circle represents a specific type or population of T cells. So for a stage 0 inactive Sarc sample, basically this is a sample and a phenotype that is mild. We are not seeing any of those white circles, meaning we're not really seeing any examples of clonal expansion. And clonal expansion, in this case, we would interpret as an ongoing or active immune response. So there's really no signs of inflammation occurring for this sample. If we move to a different phenotype, stage 2, again, we're not seeing any of these white circles showing up. So again, no example or no evidence of clonal expansion. However, if we move to a sample that has been characterized as being progressive, stage 2 progressive, meaning more severe disease, or at least active disease, now we are starting to see some of these white circles showing up. So this is giving us confirmation that there is an ongoing immune response happening. There's evidence of clonal expansion within the T cell compartment occurring. And again, this is another sample, another stage 2 progressive sample. So again, clinically, we're saying that there is ongoing disease. And based on the TCR analysis that we're able to see, we're seeing clonal expansion occurring as well. And so what we're able to conclude when thinking about the data coming off of this TCR analysis is that progressive disease, or disease that we know that is active, does seem to be correlated with clonal expansion, whereas more stable disease does not show us any examples of clonal expansion. Moreover, when we compare TCR usage, so the V genes that are being clonally expanded within different samples, we're not seeing the same genes being used, meaning there doesn't seem to be overt evidence for a single antigen or a single pathogen driving this clonal expansion. This is very interesting for us in the sarcoidosis world, because one of the major questions we have with sarcoidosis, again, is what drives this disease. It is a disease of unknown etiology. So there are ideas, because it is a granulomatous disease, that there might be dominant pathogens that drive this disease that we just haven't found yet. Since we're not finding evidence for specific V genes being used, it would argue that this disease is more complex and environmentally driven, which is what a lot of us think is happening with sarcoidosis. So TCR, ion TCR sequencing, we were able to apply to sarcoidosis very easily. These graphs that I'm showing you were very easy to generate. It's very user-friendly, and it gives us a tool that is very hard to do in any other way for this amount of money to produce. So it gives us a tool where we can pull out and identify and visualize specific clones within the entire T cell community, cell population. So it really has helped us identify active disease. So next I want to change up the case study. And so now I'm going to tell you about some work that Mimi Saavedra and Lee Reinhardt have done at National Jewish Health working with cystic fibrosis. So cystic fibrosis is a chronic respiratory disease. One of the issues with CF is that it is characterized by repeated acute pulmonary exacerbations. And these acute pulmonary exacerbations over time lead to worsening lung function. And one of the primary reasons is that after the exacerbation, a lot of the patients do not return to baseline lung function, so their lung function decreases due to these exacerbations. So Lee and Mimi came up with this hypothesis that T cells are getting differentially programmed over time, and that within the memory T cell compartment, you're seeing a dysfunction, an immune dysfunction that occurs. So that with these repeated pathogen infections that drive a lot of these pulmonary exacerbations, you're getting this response that does not clear. So normally pathogens are cleared efficiently, and because of the presence of memory, you get protection from subsequent infections. In the context of CF, that doesn't occur. And so what they wanted to do was look at memory cells within the PBMCs and examine the function of those cells and try to understand why it doesn't seem that patients are protected. And so they set out to do this clinical research. One of the things that they initially found when looking at the memory T cells is that they seem to have a functional bias. So here they took peripheral blood derived memory T cells, CD4 positive T cells. They stimulated them in vitro and then asked what are they doing functionally. And they saw this Th17 and Th2 bias. So Th2 being identified by IL-4 production, and then they also looked at IL-17 production. So they compared CF subjects who were sick versus samples from CF well versus normal controls. And one of the things that you can, I think, easily see is that there's a lot more production of IL-4 and a lot more production of IL-17 from the CD4 positive memory cells derived from the CF sick subjects versus the well subjects or normal controls. So it really led them to this question of, okay, what's happening in the context of the inflammatory reaction? Is there a specific set of T cells that are driving this inflammation that we know leads to decreasing lung function over time? So they went ahead and did the ion TCR sequencing. And this is another type of graph that you can, again, easily produce with the tools that Thermo provides. So here, what you're looking at are graphs that are now showing the unique combinations of V and J genes that are used within individual clones. And again, it's a similar readout. So white dots or white squares here are going to show highly expanded clones. So with the CF subjects, it's the two top graphs. You can see these white squares that are showing unique combinations of V and J genes that are highly expanded. And if you compare that with the normal sample below, you'll see that the normal sample has a lot of this white and gray spread throughout, whereas with the CF samples, there's more defined regions of the white and the gray. Essentially, this is that same concept of evenness. In the normal sample, you're seeing more even distribution of these different use of the V and J genes, whereas in the CF, it seems to be more restricted. So what Mimi and Lee observed and what they concluded, that they were doing this kind of analysis, they were able to actually identify dominant clones that were tracking with CF inflammation. And when thinking about the amino acid motifs that are coming from these sequences, they're wondering and starting to ask questions about, can you use that to identify the bacterial antigens, the pathogens that are driving this repeated and chronic inflammation in CF? When they compared the CF sequences versus the normals, they were able to get a greater number of motifs from the CF samples. And when looking at those motifs, there were more shared motifs in common in the CF samples versus the normal samples. And the large majority of those samples, of those motifs that were common in the CF samples, they didn't even see any of those in the normals. So again, they got really excited by this because it's starting to really give them a handle and a window into the specific T cell clones, the specific T cells that seem to be involved in this chronic inflammation that's occurring that drives down lung function. So some of the questions that they were then able to ask and start to think about is, do the expanded clones, these specific clones that they're seeing in the CF samples, do they translate to greater inflammation, and do they translate to worsening lung function? And so they can use things like that are available, like glyphs, to try to identify shared motifs. Again, this idea of convergence. Are there T cell receptor genes that may be differential genes, but that seem to be identifying the same antigenic peptides? And so in doing that kind of analysis, and again working with the tools of Ion Reporter, they can compare the motifs that are being seen in the CF samples versus the normal subjects. Here they're doing principal component analysis, PCA analysis, and again the CF subjects are clustering separately from the normal samples. And again, doing the same analysis, they saw a lot of the same types of things, that they're seeing that the CF motifs are shared, and they are not within the CF samples, but they're not shared with the normals. So moving forward, they're really excited to use this technology as a window into the chronic inflammation that's occurring in CF, to understand what's happening in the memory T cell compartment, and potentially use this kind of research to understand, are there specific types of bacterial pathogens that are driving the expansion of specific T cell clones? And if that's true, then you can start to think about ways to block that and maybe stop this chronic inflammation that leads to this worsening lung function over time. So I'd be remiss, we'd all be remiss if we didn't think about SARS-CoV-2 and how that's affecting all of our research in anything that we do. It's something that at National Jewish Health, being a respiratory hospital, we've had to think about very carefully. So I want to briefly go over how we are beginning to apply TCR sequencing and BCR sequencing to both of these projects, again, as examples of how we're all going to have to think about this virus and how it impacts our research studies. So in the context of sarcoidosis, again, we know that it's a T cell driven disease. And if we are thinking about the molecular programming that occurs in the context of these chronic diseases, we understand that due to combinatorial diversity, you can have population changes. So different TCRs being used within the population of T cells. You have to start to wonder, how does a virus like COVID-2 potentially affect that interaction? So the viral response is going to lead to unique disease signals. Specific clones are going to be activated. How does all of that interact in the context of an ongoing chronic inflammatory disease? So this is something that we'd love to understand. And so I hope by now you've understood that the TCR sequencing and also the BCR sequencing gives you a really nice tool to reach into the immune system and identify, be able to see what's happening within T cells and B cells. So we can use this tool to start to understand how the presence of the virus may affect the function of T cells and the function of B cells in these diseases. So for sarcoidosis, we want to use TCR sequencing to identify unique clonal expansions that are happening in the presence of the virus versus not. We think that, again, because this is viral infection, that there could be changes in the T cell lineage, so the function of the T cells. And again, this may have impacts on disease progression. So we know that sarcoidosis is characterized by Th1 activity. We're also seeing examples of Th17 or even Th2 or TFH. So how does the virus impact on the functional aspects of the T cell population? And of course, there's obvious questions about how the virus may affect disease course over time. In the B cell compartment, when thinking about BCR sequencing, there's ways to identify clonal expansions. And of course, anytime we're thinking about viruses and vaccines or antibody responses, it'd be great to identify those clonal expansions, the B cells that are responding. So we'd like to use BCR sequencing to try to get a handle on that. We can also think about B cell maturation. So at least within sarcoidosis, we know that there's a presence of these TBET positive B cells, and we'd like to understand, are there specific effects on these kinds of B cells due to the presence of the virus? And one of the things that we haven't had time to discuss, but again, the BCR sequencing tool from Thermo allows you to look at progression or maturation of B cells. So looking at plasma cells and antibodies and isotype changes or somatic hypermutation for affinity maturation allows you to track all of those things. Again, we expect that will be affected by the presence of the virus. So being able to apply these tools in the context of sarcoidosis, expecting that the virus, the presence of the virus should change some of these things, having the ability to look at the T cell compartment and the B cell compartment at that individual clone level gives us a really powerful tool to understand what's happening in the immune system over the context of this chronic disease. It's a very similar situation with CF. So again, it's a chronic inflammatory disease. We know the immune system is heavily involved. So what Mimi and Lee have talked about doing with TCR sequencing is, again, to identify those novel clonal expansions. Since they are able to see some dominant clones that seem to be associated with CF, how do those clones change? Are there going to be new clones that arise because of the presence of virus? And what's happening to the clones that they've already identified? They are interested, again, in this idea of T cell convergence. So what's happening to the overall ability of the T cell population to see specific types of bacterial pathogens and antigens associated with those bacterial pathogens? So does the presence of virus change that or alter that? And of course, we all expect that the presence of virus could have an impact on disease course. The same thing in the B cell population for them as well. Since they're really interested in trying to hone in on bacterial pathogens and antigens that are driving the ongoing inflammation that leads to worsening lung function, being able to see the other side of the adaptive immune system, the B cell, and being able to track clones and hopefully work backwards to help identify specific antigens would enable them to get a better handle on what's happening with the presence of virus in CF. And so finally, I want to talk about how TCR sequencing and BCR sequencing, at least as employed by Thermo, fits in the context of these kinds of mechanistic biomarker targeting studies in clinical research. So one of the great things about this assay is it's essentially a bulk assay. You can use it with T cells, B cells. You don't have to go through flow cytometry to identify a specific cell type or specific subtype of your T cells or B cells, unless, of course, you want to do that. But it's still kind of high resolution. Again, because of this combination of PCR and next-gen sequencing, the PCR is targeting the assay to just look at the TCR or the BCR sequences. So in comparison to something like RNA-seq, if you were to try to do BCR or TCR sequencing by bulk RNA-seq, you would be wasting a lot of, not wasting, but you'd have to sequence at a really high level to get to your TCR BCR sequences. And even then, your ability to see all of the potential combinations would be really low. TCR sequencing and BCR sequencing, because it's targeted, has that higher resolution for looking at the antigen receptors. And then, of course, as many of you know, you can do BCR sequencing and TCR sequencing with single-cell RNA-seq approaches. The huge advantage of single-cell RNA sequencing approaches over Thermo's approach is that you get to sequence the entire receptor. So for TCR, you get to look at both TCR alpha and beta chains, whereas Thermo's approach only looks at the beta chain. So the huge advantage that the single cell system has, or the approach has, you get the entire antigen receptor. The drawbacks of single cell is that, as many of you know, you're only going to be able to look at a few thousand cells at a time. So you're not really getting that big picture on the entire population. And of course, it's a lot more expensive than ION's approach. So there's pros and cons of both, which is why at National Jewish we really use them as companion assays. They're really ideal companion assays. For some of the questions that we address, we're just trying to track clonal expansion, so we don't need to have both the entire antigen receptor sequence. We don't need the alpha and the beta chain. So the ION platform works really well. For those questions where we really do want to have the entire receptor sequenced, we'll do limited single-cell RNA sequencing, again, because of the limitations with the cell numbers and the cost, to identify the exact receptor that's being used, and then we can follow those up with TCR beta sequencing on a much larger number of samples to get a better idea of what's happening across a large population. So there's really pros and cons of both approaches, but we've gotten a lot of use out of Thermo's approach. And then finally, at National Jewish Health, we are a service provider for Thermo Fisher TCR, BCR sequencing, and other AmpliSeq approaches. We know that a lot of different institutions don't have access currently to Thermo ION sequencers. So if that's something that you need or you're interested in, we are a full service solution for that. Here's some of our contact info. You can contact our genomics facility directly at our email address or phone number, and there's also a website where you can get some more information. So I just want to finish up by thanking the audience for attending today and for your time. Again, I want to thank Thermo and Labroots for hosting this, and I'd like to open it up to any questions the audience might have.
Thank you, Dr. O'Connor, for your informative presentation. We will now start the live Q&A portion of our webinar. Now, to our audience, if you have a question you would like to ask, please do so now. Just click on that Ask a Question box located on the far left of your screen, and we will answer as many of your questions as we have time for. Okay, let's get started. We have some great questions already coming in. Dr. O'Connor, let's start with this one. How would you compare this method with single-cell sequencing?
Yeah, that's a great question, and that's one that we get all the time at our genomics facility. So researchers come to us with with different questions that they want to address, and one of the first questions they have is, what's better? Should I be doing single-cell sequencing, or should I be doing this more bulk approach? And it really does come down to the specific question that you have. If it's a question of if it's an immunology question where you really do want to understand what the entire antigen receptor looks like, then single-cell RNA sequencing is the best way to go, because you get to have both chains being sequenced. However, for most of our users, we wind up going more with the Thermo approach, and honestly, one of the big reasons is cost. And the other reason is the ability to look at a large number of cells. So you can literally take millions and millions of T cells out of peripheral blood and apply the Thermo approach and get a large number of the clones identified in your sample. Whereas with single-cell RNA sequencing, even with modern techniques with hashtagging, you're only looking at a few thousand cells at a time. So it really does become a trade-off of, do you really need to know the entire sequence for your antigen receptor versus do you want to look at a large number of cells in a population? For questions of clinical research where you need to look at lots of numbers of samples, Thermo's approach really does become a really powerful tool, because you can look at lots of cells and because of the lower cost. For a lot of our more basic science types of questions, that's where we see more use of the single cell.
Thank you, Dr. O'Connor. Can you talk more about how you would use TCR-seq in your analysis for sarcoidosis disease progression? specifically in terms of phenotyping disease course that gets worse versus gets better?
Yeah, that's one of our favorite questions, and it's the one that we think about a lot. So again, with sarcoidosis, it's a disease of unknown etiology, and it's variable disease course. And also we see that a lot in autoimmune type or inflammatory type chronic diseases. So everybody would love to have some biomarkers or some handle that within the immune system that would help us understand why there's differential progression or differential disease phenotypes, and ultimately why disease course looks so different. So we've been tracking T cells and B cells in sarcoidosis through things like RNA sequencing. My lab is specifically interested in epigenetics, so we look at histone modifications by chip sequencing. And through those methods, we've been able to, at a bulk population level, understand some of the molecular programming that's happening inside of the cell. So whether a cell seems to be programmed more towards a Th1 type of function or a Th17 type of function, and then we try to relate that to what's happening in terms of disease course. But none of that type of analysis, RNA-seq or ChIP-seq, none of that type of analysis really gives us a handle on individual clones or individual T cells and what's happening within the T cell population. And because we know that a hallmark of adaptive immunity is this idea of clonal expansion, the observation that when an immune response occurs, you see this expansion of a specific clone, which then translates into a specific unique combination of T cell receptor or B cell receptor genes. Being able to observe that expansion of those T cell receptor and B cell receptor genes allows us to understand what's happening actively in those two compartments. And so what we're doing now with T cell receptor and B cell receptor sequencing in sarcoidosis is trying to understand what is happening in B cells and T cells over time. And how does that correlate with some of these differential molecular programming that we're seeing? So some of the questions we're trying to ask are, do we see changes in TCR genes being used? Essentially, are we seeing different kinds of T cell clones associated with different T cell functions? So are we seeing examples of clones that seem to be associated more with Th1 function or more with Th17 function? Are we seeing specific clones arising that seem to be associated with different disease progression? And I think some of the evidence I showed you today is that, at least early on in very preliminary analysis, we are seeing signs that specific clones do seem to be associated with progressing disease, or at least active disease. So the way that we're thinking about using TCR is applying it in the context of these other assays, using assays such as RNA-seq or ChIP-seq to understand the molecular underpinnings of the function that seems to be associated with worsening disease, but then understanding, does that seem to be associated with specific T cell populations through this TCR analysis. We're doing something similar with B cells. We do see evidence of a specific B cell function that seems to be associated with sarcoidosis, and so again, we'd like to understand, is that broad across different kinds of B cells, or does it seem to be associated with specific types of B cells? And that's a line of research that we're just starting to get into. So we think of TCR and BCR sequencing as really a nice way to sort of reach into the immune compartment and try to identify specific activity at an individual clone level.
Thank you. Let's go with this next question. What kind of dedicated bioinformatic resources do you need to perform this data analysis? Yeah,
so I hope that I was able to give everyone the idea that this is a really easy system to use. Again, I want to say I'm not trained in bioinformatics or computational biology, and all of the data figures, or a lot of them anyway, that you saw today, I generated, and that says a lot. So we have really great bioinformatic resources and computational biology researchers here at National Jewish. We have a whole dedicated facility for bioinformatics. We have a supercomputer cluster on campus. So we have really beefy capability, and you really don't need it to do this kind of analysis. Thermo Fisher has provided a really amazing, very intuitive, very user-friendly tool that is fairly seamless with the assay. We have done some TCR sequencing for individuals outside of National Jewish and have been able to interface with them through the tool, and they found it very easy to use, very intuitive. So really, if you're a lab that doesn't have access to lots of bioinformatic tools or individuals, and you don't really know anybody who's a computational biologist, you can still do this type of analysis. Thermo's really great at providing you a walkthrough with the online tools and what's available. So I really do want to encourage everybody, if you don't feel comfortable in the computational space, that's really not a barrier to doing this kind of analysis.
Can you compare and contrast the use of DNA versus RNA for the assay?
Yeah. So we've had, I have to say, most of what we've done thus far has been using RNA as our input, and we've had really great luck going down to very low amounts of input. And generally, in the context of clinical research, you're taking your samples and trying to do lots of different things with them. So it really fits in well in that context. So we typically are taking our samples and trying to pull off bulk RNA-seq and maybe ChIP-seq. And so being able to use a low amount of material going into this analysis is one of the huge advantages that we find. So that's on the RNA side. We have had a couple of examples, and again, I think a really good example of this would be somebody who's sitting on a lot of FFPE samples. Those samples tend to be, when you isolate the nucleic acid, tends to be really degraded material, and usually for next-gen sequencing, that's really difficult to deal with. The AmpliSeq system handles degraded material really well, again, because it's this combination of PCR and sequencing as the readout. So it handles that really well, and then that's really a context where you'd want to use DNA, and that's what we've done in the past, and again, it's worked really well in those cases. With DNA, you need more input material, but again, if you're dealing with, or if you have FFPE samples, usually that's not a problem. So those would be two different use cases. I know that some of the other use cases would be if you are specifically interested in cancer, and maybe you're working on TILs, tumor infiltrating lymphocytes, and you want to look at frequencies of different types of T cells, or maybe you're looking at hematological malignancies and you want to look at frequencies. That's really, again, where you want to use DNA. There's really not a great way to use RNA for deconvolution of that data. So there's different use cases. It really, I think, does come down to what your specific questions are.
Dr. O'Connor, you talked a little bit about infection-driven exacerbation in respiratory disease. Is that a big problem? And how could you use NGS-based approaches to address that issue?
Yeah, really good question. So for a lot of the diseases that we see in National Jewish, these respiratory diseases, one of the huge problems that we all face are these exacerbation periods that lead to worsening lung function. And a lot of times, these exacerbations are driven by infections. So in the case of CF, where they're really strong, robust infections, they're fairly easy to see, quote unquote. And so doing TCR-BCR sequencing allows you to see the immune response. So essentially track the infection by tracking the immune response to that infection. In other cases, we are dealing with exacerbations where there's suspected infection, but it's really difficult to track. And so that's a case, again, where AmpliSeq works really well. So we had a project that was funded by the Bill and Melinda Gates Foundation looking at MTB, tuberculosis. And we were able to use an AmpliSeq approach to track the presence of TB in different samples over a treatment course. And we tried to do this with other typical methods such as 16S qPCR. And really the AmpliSeq approach, again, because it's that combination of PCR and sequencing, amplified the signal to a point where we were able to get better resolution, or at least equal resolution to what we were able to get with 16S qPCR, but with the huge advantage that now we could track lots of different gene expressions specific to TB, which allowed us to ask a lot more questions about what was happening to the pathogen in the context of the disease course. So there's lots of different, the power of AmpliSeq and the approach that you can take with NGS to ask questions about how do infections contribute to disease and disease course over time. The resolution you get with NGS it really opens up a lot of new types of questions.
Thank you, Dr. O'Connor. Now we have time for a few more questions, but I do want to let our audience know that any questions we are unable to answer live today, and additionally those that are submitted during the on-demand period, they will be addressed via the email address you provided at the time of registration. So we have time for a few more. Let's go with this one. Why would someone choose the short read versus long read version of the assay?
Yeah, other good questions. So we didn't get a whole lot of time to talk about it, but yeah, there is a short read versus a long read version for both TCR and BCR sequencing. So the short read version, again, would be great in a situation of FFPE where you suspect that you're going to have a lot of degraded sample and you're not sure about how much of the intact gene you're going to have, gene expression you're going to have. Or DNA, intact DNA recombination that you're going to have. So the short read assay would be a really great application for that. In contrast, if you are a B-cell immunologist or if you're dealing with a clinical research project that has a B-cell component to it, and you're interested in looking at class switch recombination, so the isotype switching, that's really where you're going to want to look at the long read assay, because you're going to get the entire VDJ segment plus the constant region, that isotype switching. So again, they're really they're two nice options to have, and it really does come down to what's the material that you're working with, what's your question, how much information you're looking for. If you really just are trying to track clones, the clonal expansion, or observe them, then you probably could use the short read assay. If you want to understand more about the immunobiology, such as class switch recombination, then the long read assays are a really good option for you.
Thank you. Would you expanding on how you use different tools to examine molecular programming of immune cells in the context of respiratory disease course, specifically in terms of NGS approaches such as TCR-seq or RNA-seq? Yeah,
so I think everybody these days is doing some sort of NGS in their research, and we've been involved in different NGS assays now for quite a while. One of the things that we are starting to understand is that a lot of these assays really do feed off of each other. So if you do something like RNA-seq and ChIP-seq, we start to think of ChIP-seq looking at histone modifications as the programming, and RNA-seq is the readout of that programming through transcription. So those two assays can really feed off of each other and allow you to ask some really interesting questions of immune cells in the context of different diseases. So TCR-seq and BCR-seq, come along and add another component to those kinds of assays. Again, we are using things like ChIP-seq and RNA-seq to try to understand the molecular programming in the context of chronic diseases over time in these memory cells. But understanding what's happening to the individual cells, the clonal cells, the individual T cells or B cells that express a unique B cell receptor or T cell receptor, gives us another layer on top of understanding that programming Because if we see programming changes in the context of a disease course, it'd be great to understand if the T cell compartment and the B cell compartment is changing as well, or if it's the same kind of cells and they're changing over time. Those give you two very different scenarios and gives us a really powerful way to start thinking about what's happening to these chronic diseases and ways that we might be able to think about intervening in those inflammatory courses.
Thank you, Dr. O'Connor. And we have time for one more question. And I couldn't end this webinar without touching on this subject. As we all know, SARS-CoV-2 is overwhelming the research community currently. How do you think it will affect immunology and disease research approaches? And how can we use NGS-based approaches to help study the impact of the virus?
Yeah, that's obviously a really poignant question. And, you know, I think Thermo has come out with an AmpliSeq tool that allows people to sequence the SARS-CoV-2 genome. Tools like TCR BCR sequencing, you know, obviously allow you to track the immune response. So, you know, I don't think any of us have a really good sense of how the presence of the virus in the context of these chronic diseases, what's going to happen in terms of disease course. But obviously having these tools, being able to understand how the virus is changing with an individual through perhaps sequencing the viral genome with AmpliSeq, or understanding how the T cell compartment and B cell compartment is changing in the context of the presence or absence of virus. You know, obviously those are enormously helpful tools to understand, how are we going to have to change the way that we think about the way that we conduct our research projects for these different diseases. So just being able to see those changes, obviously that's kind of like the first step in trying to understand how we move forward.
Thank you, Dr. O'Connor, and thank you for this informative Q&A session. Before we go, I do want to thank our audience for joining us today and for their interesting questions. There were quite a few questions we were unable to get to, and additionally, those questions that come in during the on-demand period, we will make sure that these are addressed via the email address that you provide at the time of registration. I would once again like to thank Dr. Brian O'Connor for his time today and his important research. We'd also like to thank Labroots and our sponsor, Thermo Fisher Scientific, for underwriting today's educational webcast. Today's webcast can be viewed on demand, and Labroots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. That's all for now. We hope to see you again soon. Have a great day.